Hello everybody. Reproducibility is indeed a worldwide problem. Recent publications in major research journals have jointly addressed this issue. Julio, a researcher at the UQ, will briefly go through the problem and will provide potential solutions.
Yes, indeed, lack of reproducibility is the major cause of delay in new discoveries to come. A recent report in Nature Research has assigned 6 factors as clear threats in life science research. Unfortunately, some of them can't be tackled in a retrospective manner, but others can. This is why we have developed STEM ID.
Can you tell us how STEM ID works and how this could help to our scientific community?
Yeah, sure. So one big problem we face in stem cell science, and more in a broader context in life sciences, is directly related to cell identity. In other words, we scientists receive cell lines from collaborators all over the world, and often the cell lines do not behave as previously reported. So at STEM ID, we are developing a tool that will allow to compare datasets from RNA-seq experiments performed in different publications using the same stem cell lines. And this will give us a perspective on whether the real problem of reproducibility comes from the fact that certain cell lines claimed to be used in a given laboratory is likely being misidentified or perhaps cross-contaminated with serial positivity.
So, do you think this would explain the contradictory results that some laboratories have?
Yeah, that's right. It may simply be a matter of using cell identities that people assumed were right, but in fact weren't.
So now here I am with Jeryn, an UQ experienced bioinformatician who has developed the algorithms to assess the cell identity of the stem cells. Can you tell us something about that? Thank you for being here, by the way.
Thanks for having me. So we're developing an open source pipeline in R to measure how these cells have changed over pseudotime, and measuring the distances and quantifying it so scientists can know whether or not their cells are actually the right stem cells. So this is Stem Cell ID. This is a place where lab scientists all around the world can check if their stem cell cultures match the identity of their original stem cell cultures. So say if Julio wanted to check if his H9 cells match the original, he can simply fill in his details and submit the expression file of his cells. Here, Stem Cell ID will process the data and output, among many things, a PCA plot such as this one. We can observe the separation in data between the many different studies of the same H9 cells over the years. Stem Cell ID will also output the distance between Julio's dataset with other datasets, including the original. We hope that in the future, we can not only incorporate transcription data, but other omics such as the proteome, metabolome, epigenetic markers, SNPs, and more. We hope that our tool can push scientists to think more about reproducibility so that their findings translate well into a clinical setting. We hope that Stem Cell ID can jumpstart a process to develop a method to account for drifts in stem cell lines' signature IDs. Together, we can improve the reproducibility of stem cell studies across the globe.